Okay, my computer is a little slow because the lecture is still, the program is still being made from the lecture. So just bear with me for the first 10 minutes of the class probably. Are there any questions? All right, if there's no questions, let's go ahead and begin. Share my screen. There was a question. If I can close this. Don't need that yet. There was a question about how many things you can submit. I've got unlimited allowed attempts for a submission. So you can easily put 2 files in this one link. And sorry, I need to break that into 2 links, but it's too late now. So we'll go with putting 2 files in that one link. All right, I guess I can close that. Let's go back here. Remember, you can find the lab in the Canvas page under week 1 lab. And it's going to be today, module 1, ubiquity of organisms. And for some reason, I've got worksheet under one link. It's for both lab 00 and lab 01. Let me remind you, in the lab, you need to finish the plagiarism tutorial and take the plagiarism quiz by this Saturday. Although it's only 20 points, if you don't score 100% on the plagiarism quiz, then you will get 0 on the infectious disease project. And that's worth 100 points, so you don't want 0. So get that done. And then the other thing I need to remind you of is that it's right here. I think it's here. You need to submit lab module 00 by Friday at 8 a.m. Otherwise, you're going to be dropped from the class. I'm going to use this submission for determining who's attending the class, who's registered and who's attending. And if you're not attending, you will be dropped from the class. And I hope you all are attending, because we do not have that many people in this class. And so I'd hate to drop anyone. Let's see. There, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Only 12 students. And you want at least 13 for the class to run. They won't cancel the class now, but they were thinking about canceling the class like a week earlier. All right, any questions before we begin? If not, we're going to cover lab 01 today, ubiquity of microorganisms. Remember, take the plagiarism quiz by this Saturday, 11:59 p.m. You must get 100% on it. Turn in the lab 00 safety training by 8 a.m. Friday. And then another thing you should do is read the infectious disease research project. It's called the project instructions. It's under Canvas for next week, meaning week 2. I think I can show you that. Why do I have that there? What is that? My computer is overloaded at the moment, so anything I don't need, I should close. There it is. Read the infectious disease research project. I will discuss this next week, and you will be starting the infectious disease research project next week. I will not take infectious diseases, so don't send me an infectious disease until Tuesday. All right, let's go on. I think we're done here. So I'm going to close this. Like I stated, I will be available to answer questions till about 9 o'clock or the last student logs off. When the last student logs off, I'm going to log off as well. But let's start with lab module 1. I should have a lot of windows open here. We'll eventually need that one. I think that's the one I'm on less lecture from. Okay, exercise. All right, any questions? Yep, this is the one. All right, so lab module 1, ubiquity of microorganisms. You do need to do some reading before you do the lab. Page 157 to 162, and then 162 to 162 to 163. You'll be reading about culture media and obtaining pure cultures. There are some video clips in this lab, and so you'll need to watch those as well. If you complete this lab, the learning objectives are to know that microorganisms are almost everywhere, describe the purpose of culture media, be able to describe what is colony abundance, colony diversity, colony morphology, be able to define the following terms: what is inoculation, ubiquity of microorganisms, what is a sterile tube or sterile media, what is isolation, what is incubation, what is in vitro, what is culture media, and there's different types of media. There's a broth, and then there's a solid plate, which uses agar. There's also a slant, and there's a deep. I should probably get my paint out to show you these. What's a fastidious organism? What's the non-selective media? And what is your ubiquitous colony? Any questions about any of that and what we're going to be covering? All right, as you know, microorganisms are organisms that are too small to be seen with the naked eye. This does include viruses and prions, which are special because they don't have cells. And it does include helminths, the worms, even though the adults are, or the adult life stage is not usually microscopic. All of the helminths have certain life stages which are microscopic. And these are usually important because that is the stage which usually infects humans, causing disease from the helminth. You should know that the phrase ubiquity of microorganisms refers to this concept that microorganisms are essentially everywhere. In this lab, we're going to investigate the abundance and diversity of microorganisms in common environments. We'll also learn some basic lab protocols used with using a solid media, how you label lab cultures, procedures for disinfecting the lab benches, and procedures for washing your hands. The culture media, also called a growth medium, you will be, if you were going into the lab, you would get one with the sterile laboratory preparation that contains a variety of nutrients that are required for growing microorganisms. So if you had it in a tube, it would be sterile, and usually it's sterilized by putting it in the autoclave, which we'll talk about in a little bit. I think it's in our next lesson, lecture lesson. Culture media may be liquid. These are called broths, and they're commonly used in a test tube. We often add a solidifying agent to the broth, such as agar, to make it a solid media. Agar is a gelatinous substance extracted from seaweed, and it's used just to thicken the broth and turn it into a solid media. Okay, any questions about any of that? There's some videos for you to watch on preparing a pour plate. And you should answer the question. I'm not going to quiz you on it. You should know the answer to the question. Why must a culture medium be sterile prior to it being used? It would be worthless using one that is not sterile. All right, so this is a liquid broth, and it will be flat on the tube. These are 3 different tubes. This is a solid media, so is this one. And that one's just flat across. This we call a agar deep. This we call an agar slant because the media is slanted. Any question about any of that? Agar is also grown besides in, used in petri, not petri dish, in test tubes. It's also used in a petri dish. And that one, it's hard to see that that's solid, but that one you can see is solid because it's whitish in color. Most of the agar you would use in a lab, it would be in a petri dish, but occasionally you use test tubes. Broths are almost always used in a test tube. We're going to talk about media today, and there's one type of media that we're going to talk about today, and that is a general purpose media. It's non-selective in function, meaning it allows almost everything to grow. It's usually complex in composition, and it supports the growth of most non-fastidious organisms using peptone and yeast and beef extracts. A fastidious organism is something that does not grow easily, and it requires additive to the media before it will grow. Like Streptococcus pyogenes would not grow in a general purpose media because it is a fastidious organism. It needs certain additives before it would grow. There are 3 general purpose media that we're going to talk about. And let's talk about the 3rd one first, nutrient agar. It's a very simple media that has, of course, agar to make it solid. And then usually just beef extract and tryptic soy extract. That's soybeans ground down to protein. And then it's partially digested. You don't need to know that. A very simple and cheap media to make. And we use it a lot. Nutrient agar with yeast extract has yeast extract added to it, and that makes it a little richer. And we usually don't call it nutrient agar with yeast extract. We call it NAY media, meaning N-A-Y-E for the first initial of each of these words, nutrient agar with yeast extract. And then tryptic soy agar, which is frequently abbreviated TSA. These are all general purpose media, which are used to simply grow many different organisms. And we use it because they're cheap and many things grow on it. If you saw that window pop up, that means the lecture lesson, the file has just finalized on my computer. So my computer will now work a little faster. There are other types of media, but we're not going to talk about them in this lab. In this lab, we're only talking about general purpose media, and the purpose of them is just to grow many different organisms cheaply. Okay, they do not grow all organisms. No fastidious organism will grow in a general purpose media. And of course, they're non-selective, meaning we can't grow certain organisms and then select against other organisms. You should be labeling all of your tubes and petri dishes in the lab. Everything should be labeled, and the correct way to label them is always include your name or your initials, put the date on it, add the type of medium like May. The incubation temperature is optional, and then you should put the inoculum, what you're trying to grow on the medium. So this plate shows you, done by an instructor, Travis. It's growing E. coli. There's the date. I can't read the year, but this is many years ago. The media is TSA, tryptic soil agar, and it was grown at 37 degrees Celsius. You may add a piece of tape if it's a test tube. Generally on a petri dish, you write on the bottom edge of the plate so it doesn't obstruct your view of the plate if you're looking at the bottom of the plate. But you always write on the bottom. I'll explain why that is in a minute. You need all this information so that if the plate is found, we know whose it is, when it was started, what type of media it is, and what it's growing. And the temperature is optional. All right, any questions about any of that? All right, in inoculation is when you put something in the culture media. So it'd be bacteria or whatever you're trying to grow in the media. So putting it in is called inoculation. And we'll talk a little bit more about that. Incubation is when you then allow it to grow. And usually we allow our microbes to grow in an incubator, which is a place for growing the microorganisms. Complex incubators can control the amount of oxygen, the carbon dioxide, the temperature, the humidity, and then of course the time. Usually incubators in our lab only obviously control the time. You decide how long it grows, and the humidity and the temperature. We in the microbiology lab no way to control the amount of oxygen, and very little way to control the carbon dioxide. There is a way to control the amount of carbon dioxide, but we're not going to discuss it. Let me show you an incubator. There we go, there's an incubator. This is a complex one because it has dials there. All right, because if you put a plate in the incubator, which is warmed, and there's agar on the bottom of the plate, the moisture from the agar will come out of the agar and then precipitate on the bottom of the lid, forming drops, which will then accumulate and then drip down into the culture. It may not contaminate the culture because the lid should be sterile, but it does increase the chance of contamination happening, but it would ruin the culture because instead of having isolated colonies growing, everything will just mix together and then grow all in a mass. For that reason, when we put plates in the incubator, we don't put them in right side up. we put them in upside down. Any question about that? Why you put them in upside down is that there's no auger on the bottom now. It's all on the top. And if moisture comes out of the auger, it will start condensing on the top, which is the auger. And then the auger will whip the moisture, so droplets will never form. And even if they did, they would drop onto the lid. We don't grow anything onto the lid, so that wouldn't be a problem. So everything that's grown in the incubator in a plate is put into the incubator upside down. And then an advantage to putting things in upside down is we're writing on the bottom of the plate. So that will be up so you can see it if you put it in upside down. Any questions about any of that? All right, when we look at the colonies growing on a plate, bacterial colonies tend to have a rather smooth textured surface and distinct edges. In comparison, fungal colonies tend to be furry or hairy looking. So you can actually look at the colony and tend to determine, is it bacterial or is it fungal? Each colony is made from not one cell, not 10 cells, but tens to hundreds of millions of cells. And they may all be descended from one cell that went through division to 2 cells, 4 cells, etc., until you get at least 1 million cells. You need about 1 million cells before you can see the colony. Let's go ahead and take a look at some colonies. Blow this up a little. Here you can see this colony is distinct. The edges are smooth. Distinct edges. That is a bacterial colony. This edge is not distinct. It's hairy or furry looking. And these are fungal colonies. Any question about any of that? All right, we have a little video for you to watch on binary fission, how most bacteria cells grow and reproduce. Literally, the cell just splits in two. All right, when we're talking about colonies, they have different characteristics. And we can look at the colonies and learn some things about the species from the different colony characteristics. Different colonies have different characteristics. I should say different species of colonies have different colony characteristics. So you can have a shape of the colony, and the shape can be circular, it can be rhizoid, it can be irregular, filamentous, or spindle. The margin is the edge of the colony. We call this entire, that's the round, smooth edge. It could be undulate, lobate, curled, rhizoid, or filamentous. And if ever I'm asking you, is the edge of the colony undulate, lobate, or curled, I will never ask that because it's too difficult to distinguish between these two. But I may ask you, is it entire edge, or is it undulate, lobate, or curled? Or is it rhizoid or filamentous? But I'll never ask you, is it rhizoid or is it filamentous? Because those are just too difficult to separate. Elevation is how the colony looks when you're looking at it from the edge. Colony can be flat, it can be raised like a mountain. What's that term called? I can't remember. Raised. It can be convex, it can be pulvinate, that's extreme convex, or it can be umbinate, meaning looking like a mountain. I would never ask you, is the colony convex or polyvent, because it's too difficult to distinguish between those two. But I could ask you, is it umbonate, convex, or flat? Okay. Size of the colony is the size of the colony. Punctiform is smaller than the point of a pencil or a pen. Small is about the same size as the point of a pen or a dull pencil. Large is about, moderate is about the size of the eraser or the cap of the pen. And then large is beyond the size of a pencil eraser. Any question about any of that? You can look at the colony and determine, is it smooth looking or rough looking? You can look at its appearance and determine, is it glistening, shiny, or is it dull? You can look at the colony and determine, is it pigmented? Pigmented means it has color, like purple, red, yellow. If it's non-pigmented, it does not have color, and it could be cream, tan, or white, or off-white. And then you can look at the optical properties of the colony. Is it an opaque colony? You cannot see through it. Is it translucent? You can see through it, but it's not clear what's underneath the colony, meaning it's not like looking through glass or plastic, clear plastic, translucent, like looking through slightly muddy water. Or is it transparent? You can see crystal clear through the colony. Most colonies are opaque, but some of them are translucent. Let me see if I can find. Yeah, all of these look opaque. I don't see any that are translucent. And obviously there's none that are transparent. Transparent ones are hard to see. You can usually see them. That one might be translucent. Right here. That one might be, but I don't think it is. I think that's okay. Now, the good news is, generally speaking, I will give you this table if I ask you a question about the colony characteristics. Any question about that? Oh, here we go. There is a translucent colony. That one's even better. This one right there. There's two colonies here. Actually, there's 3 or 4 colonies here. So the big one is translucent. This one in the center is opaque, and it's a different colony. And that's probably a different colony as well. Oh, trying to shrink this. A circular colony with entire margin. This is an irregular shaped colony. And I would say the margin is irregular. Is that what it would be? It's not enough to be undulate, not enough to be lobate, not enough to be curl. I'm not sure what the margin of that one would be. Blow that up. All right, when I see it in larger fashion, it's probably lobate. Can I see more than just a few? All right, any questions about colony characteristics? So when we're talking about colony abundance, that's simply the total number of colonies on the plate. I don't know, there's a lot of colonies there, so I'm not going to attempt to count them. Like the abundance of cats in this picture is 1, 2, 3, 4, 5, 6, 7. 7 cats are abundant in this picture. That's the abundance of wild cats. The diversity is the number of different kinds of colonies on an agar plate. And you should use the colony characteristics, like the color, the shape, the texture, etc., to identify between different colony types. For the diversity of cat species on this plate, there are 2. The diversity is 2. There are cheetahs over here, and there are lions over here. This is too difficult to determine how many diversity there is, but let's come up here. It's a little easier. Roll this up. Obviously, there's 1, there's 2, there's 3. All of these yellow may be the same, but that would be 4. That little white one would be 5. That may be the same as that one. This little white one may be 6. That white one could be the same as that one. That's different, so that's 7. That may be the same as this one. So there's at least 7 different colony types. So the diversity of this plate is around 7. And if I were to ask you for the diversity, as long as you're close, it's okay. So if you say 10, I will allow it. If you say 5, I will allow the count. However, if you say 3, and I say 1, 2, 3, 4, sorry, there's more than 3, okay? And if you say something like 20, I would look at that and say, no way. All right, so you do have to be close on your diversity. On your abundance, you can actually count the exact number, although it'd be difficult counting the number of colonies right there and there and there. But you could count and get the exact number. Once again, on the abundance, as long as you're close, it's fine. So the abundance here would be, something like 50 to 200. It's probably 50 to 100. I'm not going to do an actual count, but any questions on abundance and diversity? All right, there's some terms you should know. And then you work on the laboratory. There's an interesting picture of somebody's hand on they touch the agar plate, and there's different organisms growing from their hand. You can skip the references. So then work on the questions, which I don't have it here. I shouldn't have closed that. There might be questions. I thought I opened that. I guess while I'm here, I should keep that open. All right. So on the worksheet, go ahead and read the worksheet and answer the questions. For protocol 1, you're looking at a number of different environments that are sampled by inoculating general purpose culture media with the following. Procedure 1 has air, 2 has lab bench before and after disinfection, 3, sampling your hands before and after hand washing, 4, the bacteria from your lips and the bacteria from your tongue, and then 5, bacteria in the soil. For procedure 1, what's in the air you breathe? You don't have to answer this question here, but you should answer all the other questions. And procedure 1, A, B, and C, you need to answer what's the colony abundance on this plate? Do a count. What's the colony diversity? look at the different characteristics and determine the colony diversity. And then C, what step would you take to reduce airborne contamination of a sterile agar plate that you are actively inoculating? And obviously, if you're inoculating it, you have to take off the lid to inoculate the plate. So what steps you do to reduce airborne contamination of the plate? So answer those questions and then move to procedure 2. What's on your arms? What's on the lab bench? We're going to sample the table before you sterilize the table, and then after you, not sterilize, disinfect the table. And then the first question, did table disinfection work? Answer no. What's the purpose of allowing the table disinfectant to air dry? And I think that's discussed in the lab, if you don't know the answer. Move on to procedure 3, what's on your hands? We're going to sample your hands before you wash your hands and then after you wash your hands. So A is before, B is after. You need to have your hands with the same amount of moisture on them in the before and after, because if you had dry hands here and then wet hands here, the water on your hands would whip the bacteria onto the plate. So you do need to have your hands a similar amount of moisture on them for before and after. First question, does hand washing work? You can answer yes and no, and you should explain why yes or no. And 2, suggest one reason why hand washing did not result in the complete removal of microbes. You may need to look up an answer to get an answer for this. Please provide a brief citation if you look up your answer. Okay. Procedure 4, when you can kiss your significant other, what exactly are you giving him or her? What's on your lips? What's on your tongue? Okay, this is your lip plate. This is your tongue plate. Please note that I've got a note discussing the tongue plate. Okay, so you should read that note. You then incubate the plates and then look at the colonies. Which plate has higher colony abundance? You have to do a count here and here and just guess, because it'd be very difficult to count all of those and those. Why do you think the plate has higher colony abundance? Both body parts are close to each other. You probably often lick your lips. Why would there be more organisms in one place other than another? And for some reason, this is a question that students frequently get wrong. So I'm going to warn you that you have to think about the answer. Okay. There are 3 reasons why one has more cells, bacteria, than the other. And I will require that you have 2 correct answers for this one. I mean, you don't have to get all 3, but you need 2 out of the 3. And yes, you could come up with other answers that I might accept. Why was the tongue plate incubated at a higher temperature than the lip plate? So answer that question. And it states it up here, I didn't read it. This plate was grown at 37. This plate was grown at 30 degrees Celsius. Procedure 5 are microorganisms in the soil. We will sample the soil and then put in an inoculant of it on the agar plate and then allow the plates to grow. And here we have the plate growing. Give me what's the colony abundance here, and answer the question, is it even possible to accurately answer this question? and explain your answer. And then answer, is the greater colony abundance from the soil compared to the lip plate? Which one has more colony abundance? Let's see, there's one other thing. Oh, yeah. You're not actually going to do any of this lab, but we provided the pictures for you to answer all of the questions. So we've done the experiment, took pictures, and then gave you the pictures to answer the question. Because this is an online class, you can't actually do this. Any questions? If not, go ahead and start working on the lab. I will be here until 9 o'clock, or the last student logs off of the class.